All right, it's 1 o'clock. We're going a little bit over our typical plant breeding and genetics circle time. So if you need to be somewhere in a little bit, I would suggest to leave the room now versus the middle of our next presentation, although I hope that you stay for it. By the way, another note, if anyone brought food for our food drive, which is continuing today, please give it to Laura Macer after the presentations are over with today. All right, our next speaker is going to be Clinton Steketee. So Clinton is our final speaker today. He's a second-year PhD student in the Institute of Plant Breeding, Genetics, and Genomics at the University of Georgia, where he's also received his Master's of Science degree in 2014. He got a Bachelor's of Science degree in Crop and Soil Sciences from Michigan State University in 2012. His dissertation project right now focuses on the evaluation and characterization of drought tolerance in soybean. So, take it away, Clint.
All right, so yeah, thanks for that introduction, Brian. And I really thanks for the invitation to come here and share with you some of my dissertation research. I'm really excited to talk with you today, and it's been great visiting with you over the past few days here. So I know there isn't really much soybean production here in Texas, so I just wanted to give you a quick introduction to soybean, what soybean's all about. So it's actually the world's leading oilseed crop. In the U.S. in 2014, it had a $40 billion value, so it's a very important crop. In 2014, it was grown on 84 million acres in the United States, so just behind corn in terms of acreage. It's a diploid crop with 20 chromosomes. It's a C3 metabolism plant. And then in terms of what its end use is, it's mostly used for its oil and protein. So after the crushing process, the protein meal is fed to livestock, and then the oil is used for human consumption. So before I actually talk about my research, I wanted to share with you kind of a little tidbit of history about soybean. So recently there was a new historical marker that was placed in Georgia to signify where soybean was introduced to North America. So back in 1765, near Savannah, Georgia, is where soybean was actually first introduced to North America. So we put a historical marker there back in January to signify this event. So I just want to share that with you. It's probably something that maybe some of you didn't know. So, and then next fall, we're actually having a big World Soybean Research Conference to celebrate this anniversary, the 250th anniversary. So Georgia's really excited to be a part of this and host this research conference as well. So in terms of drought stress in soybean, it's a pretty significant issue that threatens agricultural productivity in Georgia, and then also in other more important growing regions of the United States. For instance, in terms of yield, it can reduce yield by about as much as 40% in really severe cases. Irrigation is expensive, and it's not always an option for farmers. So having said that, developing stress-resistant soybeans would be good in terms of trying to alleviate the impact that the drought has on soybean production. There's several different traits that people have tried to look at in terms of drought tolerance, but in terms of the genetic mechanisms that govern these traits, it's pretty poorly understood at this point. So that's why I'm going about this research. So objectives of this study that I'm going to share with you today. The first objective was to identify soybean germplasm that has drought tolerance by phenotyping some different traits, both in the growth chamber and also in field conditions. And then also the next goal is to elucidate some genomic regions that are responsible for these traits, and I'm going to share with you some of the ways I'm doing this with a genome-wide association study approach. Also, before we could do a genome-wide association approach, we needed to assemble a panel of genotypes to evaluate. So we put together a panel of 211 soybean genotypes. These genotypes came from geographic areas of the world that are prone to drought or low annual precipitation. And it also includes commercial varieties and also varieties that have already been identified as having some drought-related traits. In terms of the maturity groups, they're mostly from maturity groups 6 through 8, which are the primary maturity groups that are grown in Georgia. And then also, I'll talk about this more later, but there's genotypic information available for all these lines enabling us to do this GWAS approach. So this is a principal component analysis showing kind of the genetic diversity of this association panel that we used. Each different color represents a different country of origin. Most of the lines that we used in this association panel are PIs, or what we call plant introductions. So basically this is just showing the country of origin of where these different lines come from. So you can see in the light brown is China, China is actually the center of origin of soybeans, so that's where we see a lot of diversity in our Chinese lines. And up here is the United States, and there's a lot less diversity in these United States lines. But overall, you can see there's quite a bit of diversity, and that's what we wanted to have in terms of being able to do this association study. Also, one trait that we evaluated was canopy wilting in the field. So we did this at 2 locations, in Athens, Georgia, as well as with some collaborators at Kansas State University. And that picture on the right is showing an overhead picture using the drone of 2 of the replications of my study. So we evaluated about 200 entries at both locations. There's 3 replications of 2-row plots, so as I mentioned, the picture shows two of those replications. It was a randomized complete block design, and then also it was under rain-fed conditions, so we didn't have a rain exclusion shelter or anything like that. We relied on Mother Nature to basically give us drought conditions at these field sites. So I'll show some results of the canopy wilting in Athens. We rated these plots on July 29th after a period of about 3 weeks with no rainfall or no significant rainfall. And this was in the late vegetative growth stages of the soybean plants at this location. And this picture at the bottom here is showing kind of the differences in wilting, kind of an extreme example. I'm not sure if everybody can see it that well, but these plants here on the left are wilting quite a bit faster than the plants on the right, and that's the trait we're measuring, is basically how fast these plants are wilting. So we used a scale of 0 to 100, with 0 representing no wilting, and 100 equaled plant death. As you can see from my frequency distribution, we saw a pretty big range of responses or wilting responses at the Athens location in this past growing season. So we had anywhere from 0 to 80 in terms of the scale, and it followed mostly a normal distribution. We also did this in Salina, Kansas, and this is showing it's not quite as visible in terms of the differences in wilting, but we did see some differences in wilting at Salina as well. And these were actually recorded at 3 different times in the early reproductive growth stages, so a little bit different time frame in terms of the growth of the soybean plants. We used the same rating scale, but the ratings were quite a bit less severe than they were in Athens. But we still went ahead and used this information to do the genome-wide association studies as well. So one other thing I want to talk to you about, another trait that we evaluated, we did aquaporin inhibitor testing, and I'll go through the procedure of how this works on the next slide, but I'll give you a quick introduction to it here. Aquaporins are water transporting proteins, and kind of the sensitivity of aquaporins within soybean plants, the populations of these aquaporins, in terms of their sensitivity to chemical solutions, can be correlated with a couple of different drought tolerance traits. This work has been done by some more physiology type studies to identify correlation with these aquaporin populations to specific drought-related traits. So I'm going to talk to you a little bit about that. So one chemical that we evaluated was the silver nitrate inhibitor. And the silver nitrate inhibitor, based on the response of these aquaporin populations, a low or negative decrease in transpiration rate, which I'll explain in a couple slides, can be associated with limited hydraulic conductance, and that's an important trait for drought tolerance in soybean. So the protocol for doing this was developed by Dr. Tom Sinclair at North Carolina State, and basically what he's done is a kind of a smaller scale, and what we did is kind of ramped up the procedure to test this whole association panel instead of just a few selected genotypes. So this is the protocol. Basically, you first grow the plants in the greenhouse for about 3 to 4 weeks till they get to the V3, V4 growth stage. And then after they've gotten to that stage, you deroot the plant and put the shoots of the plants in these flasks that contain deionized water, and then wrap the top of the flask with parafilm to ensure any water that moves out of the flask is through the plant itself as opposed to just evaporation of that water. And then you place the plants in the growth chamber overnight, let them get acclimated to the growth chamber conditions, and then the next day you turn up the heat, decrease the humidity to increase the vapor pressure deficit, and encourage the plants to really transpire. And you weigh them initially, and then wait 1 hour and weigh them again and see how much water they've lost inside that flask over the course of that hour. And that's a pseudo measurement basically for the transpiration of the plant at that time. So we do that in the deionized water at first, and then we transfer the plants, the shoots, over to flasks that contain the inhibitor solution. So it's a a small micromolar solution, in this case of silver nitrate, and then we basically do the same thing. We weigh them initially, and then weigh them again after a period of time to see how much transpiration occurred of that solution compared to the deionized water. So this is how we, basically the measurement we got from doing this. We had the final weight of the flask of the derooted plants plus the flask minus the initial weight for water, so that's what TRW is. And basically the same thing for the inhibitor solution, so that's what TR inhibitor is. And this basically we were able to calculate a decrease in transpiration rate, which is shown here in this equation. And basically this is just basically how much the inhibitor solution affected transpiration compared to the water solution, or the regular water. So this is showing the response of my 200 genotypes over 7 replications of doing this, so 7 times doing it with 200 genotypes. You can see there's quite a bit of variability in terms of the decrease in transpiration as a response to this inhibitor solution. Down here, if you recall, the negative values are what are associated with this potential limited hydraulic conductance trait. So you can see there's quite a few genotypes that have kind of these low or negative values for this particular response. So then basically as a result of doing these phenotypic screens, we've identified some new germplasm of those 200 genotypes that potentially have drought tolerance for those particular traits. So we identified them in the growth chamber and also in the field. And then, as I mentioned in that previous slide, there's over 10 genotypes that had really very negative silver nitrate DTR that could potentially have this limited hydraulic conductance trait. Then there's over 20 genotypes that had really slow canopy wilting at both the Athens location and then the Kansas location. So then after we've collected this phenotypic information, we wanted to do genome-wide association analysis to see if we could identify the genomic regions that are responsible for these traits. Just as a background, we've already talked about this a little bit today, but it's a powerful tool for identifying genomic regions of interest. Basically, you use the phenotypic information you collect from, I use it from the field evaluations and also the growth chamber evaluations. And then also you need genotypic information. So we have, in soybean, we have a soy 50K SNP chip. It's a Illumina-based genotyping system where we have about 42,000 SNP markers that we can use for this analysis. And that's plenty with soybean because LD isn't quite that high in soybean. Or it is, I'm sorry, it is higher in soybean, excuse me. But then basically we also use this to ensure that the association panel was also genetically diverse. So soybean is kind of notoriously inbred and not too much genetic diversity. So we use a kind of a cutoff of 85% similarity based on these SNP markers. And that's kind of why you saw that pretty good genetic diversity in the PCA plot that I showed earlier. And then we use a compressed linear model in the GAPID-R package to do this analysis. So now let's go through and show kind of these preliminary data of these association analysis for the different phenotypic traits. So this is for the decrease in transpiration response of the silver nitrate inhibitor with 7 replications of data. As you can see, there's quite a bit of variability, or kind of a lot of noise, but you kind of expect that with it being drought tolerance. Drought tolerance is a quantitative trait, so you expect a lot of loci to be involved and important. But you do see on chromosome 15 there's a pretty significant SNP associated with this trait. And I forgot to mention, but basically these are Manhattan plots with the x-axis being a different chromosome soybean, and essentially the y-axis is the significance associated with that particular trait. So you can see there's some regions that are probably worth looking into, and then, but as I mentioned, there's kind of a lot of noise, and hopefully with more replications and more data, we can kind of hone in on some genomic regions that are really responsible for these traits. And then here's canopy wilting, kind of a similar story. Found some significant regions on chromosome 8, chromosome 20. And then actually, with the Salina data, we see a kind of a similar location on chromosome 8, so that's kind of something that we might be interested in looking at a little bit further. But hopefully we continue to do more phenotypic screenings and continue to improve the GWAS model to identify these genomic regions. So in summary, drought is a significant issue that affects soybean production. We've identified some soybean germplasm with improved drought tolerance. And then we've also identified some putative genomic regions that are responsible for these traits using the genome-wide association study approach. And I'll plan to reevaluate this association panel again in the field in the 2015 growing season. And then I'm also planning to do some studies with some linkage, some real populations to basically do linkage mapping to hopefully confirm the locations of these genomic regions. And then eventually, if I do confirm the locations, we would develop some markers that we can use for marker-assisted selection. And then I'd integrate these new germplasm, new QTL into elite lines to improve drought tolerance in soybean. So with that, I'd like to thank, obviously, DuPont Pioneer for the travel scholarship. It was really a neat opportunity to come here and to get to experience the symposium. I'd also like to thank my advisor, Dr. Zingli Lee, as well as our lab and collaborators. And then I'm also funded, my PhD fellowship is through the United Soybean Board, so I'd like to thank them as well. If we have time, I'll take any questions you might have.
Could you explain just a little bit what GAFIT is about? In R, I just wondering what that function is.
Yeah, so it's just it's a R program that you can use to basically, if you have phenotypic information and genotypic information from a plant, you can basically plug that information into this R program, and then I don't know that much about how it works exactly, just how to basically format my data to go in there, but it's essentially just a way for you to identify the significance of a particular SNP with the trait. That's what was shown kind of in those Manhattan plots, is kind of the significance of a particular SNP or that region with a trait of interest.
Sounds like you said you averaged the data of those 7 reps?
Yes, that was the average of 7 replications.
So why didn't you try and fit it into a more complex model and use the bluffs?
That's something I haven't tried yet. I think maybe that's something I should look into doing. But at this point, all we've done is just basically average the wilting data and also the aquaporin inhibitor screening data and use it with GAFIT. But that'd be something that I should probably look into doing.
Did you block it where you're doing 1 rep per time of the growth chamber?
Yeah. So we blocked it by a rep because otherwise it would have a bunch of confounding environmental effects. So we basically, each time we did the screening protocol in the growth chamber, we ran the 211 genotypes, and so that's why we had to use a large growth chamber. It was actually a walk-in growth chamber that we could utilize to do that study, and we did each set of the whole panel at once and did that 7 different times.
Clint, I have a question for you. We're starting to see more soybeans in Texas, especially like in the Rio Grande Valley a little bit. Do you have any impression that maybe with more drought research in soybean that we'll see that crop expand in this state?
I guess I can't speak to that too much, but I guess I like soybeans. I hope more soybeans are grown here. But yeah, I mean, I guess I know drought is a pretty big issue here in Texas. So I would imagine if drought tolerance is actually improved in soybean, either with my research or other research, hopefully, yeah, I guess maybe that would be something that could be utilized as a crop here in Texas.
Great. Let's thank Clint and our other speakers. All right, a couple announcements. Follow us on all of our social media sites. We would really like you to. We also would like to thank my team that helped put on all of the symposium events for all of us. Amani, thank you for all your effort. Dustin, Francisco, Smith, and especially Laura too, who helped start this symposium. We started it together back in 2015. So we're going to have some evaluations we'll send out to everybody. We really encourage you to do those for us. You can let us know what you liked about the symposium, what you didn't like about the symposium. And planning the symposium has been really a great pleasure for me. But it's a lot of work, and but you have a lot of benefits from it as well. You get to interact with our DuPont Pioneer people as well with Tabouret. You get to develop a lot of other soft skills. So if you're interested in helping plan the 2017 Texas A&M Plant Breeding Symposium, talk to one of the 6 organizing committee members from this year, and we'll let you know how you can get involved. Tabouret, do you have any last thoughts for everybody?
No, I guess thank you guys for a great start of the series this year, and again, like you said, invite everybody to continue the series along the year. You know, you guys have, our next event is in Iowa State.
Oh yes,
And we have a series of events along the first half of the year, and we promise to have a similar schedule for the second half of the year. So follow us, as you say, and follow the social media so that you can actually find out where are the other events, and keep being involved.
All right.
Thank you very much. Thank you.
Thank you very much, Tabouret. All right, that's all I have. Thank you guys for coming to the 2016 symposium.